Hi, my name is Caitlin Young. I'm a senior at Wayne State University. I'm studying biology. I currently work in an environmental consulting lab, but I plan to apply to graduate school at the end of the year for molecular genetics and genomics. I'm going to be summarizing an article from Science Daily called Genetic Roadmap to Building an Entire Organism from a Single Cell. The purpose of this informative presentation is to discuss some recently developed technologies by some researchers at Harvard University and Harvard Medical School. During this presentation, first I'm going to give you a brief summary of the research that was done at Harvard. Then I'm going to give you some general background information of the mechanisms underlying cell differentiation so that you understand the purpose of the technologies. Then I'm going to cover the 3 new technologies that the researchers developed in order to come to their findings. After that, I'll discuss some potential implications of these new methods and the new big questions following their findings. So the main idea is a team of researchers have mapped the sequential gene expression events occurring in each cell during the first 24 hours of embryonic development and zebrafish and frog embryos. So what this means is following the fertilization of a zebrafish or frog egg into a single diploid cell, the researchers were able to see which genes were turned on or off in each individual newly developed cell during the first 24 hours of growth. So essentially they can see the very first processes that cause a functional multicellular organism to form. So following fertilization of an egg, a single diploid cell is formed that will grow and divide from an unspecialized cell and differentiate into the more specialized cells that form our tissues and organs and allow them to perform their characteristic functions. So within an individual, every cell in their body contains the same genome. What causes these cells that have the same genetic information to become so different from each other is what genes they've turned on or off, which determines which proteins are being made. The researchers developed 3 new methods that work together to come to their findings. They use N-Drops, which is a single-cell sequencing technology, and they use TracerSeq and URD computational technologies as well. N-Drops is a single-cell sequencing technology. It stands for indexing droplets. It uses aqueous droplets that contain labeling molecules, and those bind to the mRNA produced by individual cells to give them a unique identifying marker for the researchers. So by labeling the mRNA, they're essentially able to see which genes are being expressed in each cell. TracerSeq is a clonal tracing method. It uses the barcodes that are made by N-Drops to show which cells came from which, or to show the specific cells that each cell gives rise to in the developing embryo. URD computational technology is similar to TracerSeq. It uses the transcription patterns to make a branch tree of specialization. This allowed the researchers to determine the spatial placement of each cell. By using all of these tools, the researchers basically found the recipe for the creation of a functional multicellular organism from scratch. Obviously, their findings were enough of an accomplishment, but they're already thinking of ways that these methods can be used to solve other problems. Some potential implications that they mentioned are perturbation experiments, regenerative medicine, where we choose the path of stem cells to heal defective cells. We can also use these methods to reconstruct any biological process where cells change their gene expression, such as cancer. So scientific collaboration is a big takeaway from the research that they did, because they had to collaborate with each other during this project, since each of them only had one of the 3 fundamental technologies available to them. Usually in scientific research, good observations lead to more questions. So now that researchers have this genetic recipe figured out, they're curious about some of the details involved, like how fate decisions are made in cells that take multiple differentiation pathways, the signals that are involved, and why different species use different cell markers. At this point, I'd like to conclude my discussion and leave you with the most important points. So, as I said before, researchers mapped gene expression patterns of zebrafish and frog embryos over 24 hours to create a genetic recipe of the development of a multicellular organism. And to do this, they used new methods of generating and analyzing data. They used ENDROP single-cell sequencing technology. They use tracer-seq computational technology and URD computational technology. And they think that these methods will be really useful for answering future questions that other researchers may have. And that's it for this presentation. Thank you for listening.